Good morning, students.
Yes.
Good morning, sir.
Yes, good morning, sir. Yes.
Okay. So, execution, what we did so far. So, we have understood what is the concept of central dogma of life. Central dogma of life. Basically, it's just the natural process of making proteins by a cell, where we have the DNA converted to RNA, RNA converted to protein is for this is for transcription, then this is for translation. Then we discussed about the theories about gene transfer, gene identity molecule. So we discussed about the Griffith experiment, then we discussed about the Avery's experiment, then we discussed about the Hershey Chase experiment, and we also have given articles about Mendel about Mendel first experiment. Then we discussed about DNA structure. We discussed the role of Rosalind Franklin, Watson and Crick, and also Murray's Wilkinson in the story. Then we discussed about what else we discussed? We discussed in detail about the replication of DNA. Can you tell me the 5 steps, 5 phases of replication? Initiation, primer synthesis, then elongation, then primer and termination. Primer cutting, removal of primers and filling of gaps, then termination. Yeah, and we also understood there are certain enzymes involved in this thing. Like initiation can be done by helicase. Helicase will unwind the DNA, whereas in primary engineering, what is the so primase working here, then we have elongation facilitated by polymerase, then we have proofreading. Okazaki fragments are reconnected with the help of ligase. Then termination is facilitated by there is some Pol 2, helicase Pol 2. So there are certain enzymes that will cut that will finish the termination. And we also understood during the initiation there are some other proteins involved, like prevention of supercoiling. So DNA supercoiling is prevented by a set of proteins. These proteins will topoisomerase and a protein. There is an involvement of one protein that is responsible.
SSB protein.
SSB, very good. SSB proteins. What is this SSB?
Single strand binding.
Yeah, single strand binding proteins, right? Yeah. So this is all about the replication of DNA. And what else we discussed so far? Yeah, we discussed the concept of proofreading. We discussed the concept of proofreading. Correct. I hope that I have one more lecture in the afternoon, right? Yeah. So I have a good amount of time to engage with this topic. So the next one I need to discuss is in the first unit we have something called as the structure of gene. What is the structure of a gene? Okay. So in order to understand the gene, okay, very interesting topic actually. Yes. Structure of a gene. So I don't know from how I need to begin this structure of gene. I just want to tell you some interesting facts before we are going through this academic depths. Fine. Listen. Gene. I hope you know the difference between DNA and gene. Can you tell me what's the difference before I discuss this one? What is the difference between a DNA and a gene?
Gene possesses it can code proteins.
Yeah, gene is something that will code the proteins. Now listen, what happened in the history of evolution means the life is always, see last bench, you are not engaged. I was very serious, I came early in the morning, I prepared for the class, and what you are doing there? Insulting me with not, paying attention to my lecture. That's very bad. And your masters, I need not to say much, you just like faculties actually almost. You can become faculties next year. And this is your attitude. Very good. Anyway, where am I? What's the problem? Genes. Yeah, the survival of genes. Keep in mind, life is always about the survival of gene. When I say survival of gene, we are saying in a broad way survival of the animals, right? Listen, I have one very interesting analogy. We know the concept of evolution. What are the rules of evolution? First you reproduce. So first you make babies. After reproduction, we got some generations. Now there is a variation. In the offsprings, there might be some variations, some black, some white. So there are some variations. That is another rule of evolution. And third rule of evolution is, let it decide which species will survive based on the resource. Natural selection. Natural selection. When I say natural selection, that depends on the particular time and the geology. It's a very, very broadest statements, right? First, let them reproduce. Let there be some controlled variations. I'm not saying can you tell me the difference between variation and mutation? What do you mean by mutation? Maybe I can say like this. We have a bad perspective about mutation that mutation is always something harmful. It need not to be harmful. All mutations are not harmful, but all harmful consequences might be a reason due to mutation. Got my point? We are not like our parents. Does it mean that we got some mutated genes? In a way, yeah, our gene was a set of new genes, but not mutated. Or maybe there are some mutations that are beneficial. So let us see in a sense of beneficial mutations. Maybe when I say beneficial mutation, these black genes, suddenly the white environment, the environment changed into dark environment, now the black genes has the high probability of survival. Maybe these genes are mutated but they have the high survival rate. Okay, at the end of the day. Now listen, the complete foresight or the hindsight of evolution is survival. How long you can survive? Your life expectancy. Genes are very selfish. They will do all these traits to maintain their survivability for the future generation. Have you understood what I said? Genes have only one motto. The gene motto is to survive for as long as period you can. As long as it can. It will do anything for its survival. Okay, for that it is doing all these things. Okay, so for me, the gene's motto is to survive for the long time. Okay, when I say long time, it can even go beyond the vehicle. When I say vehicle, do you remember I said an egg want to make another egg. Okay, egg has the information. When I say egg, that is genes. Okay, now for the propagation of these genes into the future generation, they want to do it for next 1 million times, 1 million years. So they identified some proteins. This is a genotype, and they identified some temporary proteins. Temporary proteins. Those temporary proteins can be a chicken, right? So the animals became a temporary vehicles for the propagation of the immortal genes. Genes don't have any death. And we all are temporary vehicles. So see, now we are getting a new point of view. That the concept of life became useless here. Actually, there is no depth to the information. The genes are omnipotent. They are always there. They will be there, and we are just the carriers. You know? So the genes only ensure that this carrier vehicle able to reproduce or not. That is why, very interesting thing. See, after an animal became non-reproductive or until the age of reproduction, the genes will support the animal. The genes will support the animal until it can able to bear children. Once it came to know there is no benefit of this vehicle where we can't reproduce. Now the genes won't care about us. Now the genes won't care to keep us in teenage or youthness. They will discard us because we already bear children now. We already got 5 to 6 children. So the genes are propagated and we are useless to the gene. From gene point of view, we are useless. Okay. So the age is also an outcome of genes priority. Once you can't reproduce, it don't care about us. If that is, if we have a good fertility rate, when I say fertility, reproductive time, okay, maybe to cheat the genes, we need to reproduce later in the life, not in the first or second year. Let us drag the reproductive age for more lengthy so we can live for longer time. Yeah, this is one interesting perspective. Once we know that, that's why once you can't reproduce, you will start undergoing senescence. You will start aging, and the aging rate will be drastic. They try to keep us youth for some time. After you can't reproduce, that's it. You don't care by the genes. Because their motto is just propagating themselves to the future generation. So that is called as the gene. So gene is the survival unit of evolution. Okay. It's all about the genes. Next concept I need to analyze was, see, how the genes getting their information. That is very interesting analogy. See, genes are always trapped in a dark environment. When I say gene, it is always there inside the cell. It don't have the vision of the outside world. Am I correct? The black pouches theory, where I said this black pouches and the ground is also dark. The survival rate of these genes is more compared to the white genes, because white genes will be detected by any evil predator and it will eat. Right? Now see, in this analogy, how the gene came to know that outside is dark and I will to be in the dark. It don't have the direct vision support. They are just a very single strands of DNA inside the cell. But how they are predicting the environment? How they get to know only these genes should be propagated next and these should be eliminated? How they came to know? How genes guessing the environment? Now the answer is the genes have a huge data about their ancestors. How their ancestors lived. Maybe my grandfather lived because he did the same task. Let me do the same task. That's why he is alive. So genes are completely relied on their previous data, the ancestors. Sometimes genes will try new things, that is mutation. Mutation, now you are gambling 50-50. Whether you will be survived or you will be died. The best safe game is always going with the previous data, whatever our ancestors has. Understood? Now I'm giving the concept of ancestors. The genes can only analyze their previous genes, their previous data, whatever they are carrying. Now I will show you what's happening. This is a DNA. Then there is this gene. This part is the gene. Then some DNA. Okay? Then this is the gene. Now it will ensure to maintain the record of the genes. Tomorrow there is a mutation. Tomorrow I get to know my ancestor has a black gene. So their ancestor. Everyone has black genes. So the best transcription rate will be making a black gene but not another color gene. The best survival strategy will be relying on our forefathers. Now keep in mind, the forefathers are some million years of old generations. Since the starting of life, they are having with us. We are, the future DNA is always an add-on to the previous DNA. It's just an add-on. It cannot be created from the A. It's always adding to the pre-existing DNA. Now, the DNA becomes so complicated, especially in eukaryotes. Now, the non-coding portion is called as introns. Within the gene, they have two types. The non-coding portions are called as introns, whereas the coding portions are called as extrons. Extrons are the part of DNA that will code the protein. Extrons are the part of the DNA that will code the protein. Now I'm giving you the insights of structure of gene actually. So a gene has a intron and extron. Extron will code the protein. Extron is the gene. Okay? Yeah. And my story about ancestors. So, maybe now listen. The mutation should be a controlled mutation. If it is a sudden change, definitely that will alter the previous metabolic pathways. So the mutation should have a high fidelity rate. So that is a different story, mutation, the topic. Now let us see from the point of view of the general animal. See, today we are seeing a camel, right? So how does the camel look? The camel was perfectly adapted to the desert environment, right? To the desert. Is it a spontaneous evolution? Is it a spontaneous initiation of the camel? It didn't evolve recently, because this is what their ancestors did. A camel is a living proof, or I will give you one example. Suddenly, the world changed and tsunami came, the desert became the ocean. Let us assume the desert became the ocean. Suddenly, 90% of the humans wiped out. And it took around 1,000 years to come the next generation humans. Now they are surprised by seeing camel. There is no deserts, but camel is still camel. Got my point? In future, a descendants came, humans, and now they saw the camel and they are unable to understand why this creature existed. What is the use of something that is, now it's a watery environment, keep in mind. There's no desert in the future. Now they are guessing why this camel is there. Now keep in mind, whatever animals we are seeing today, they are showing their 1 million year ago environment. It took them almost 1 million years to evolve to their current state. So every animal is a representation of a great geological time adoptions. You got my point, what I said? Every animal behavior, its characteristics, its morphology is an outcome of last 10,000 to 1 million years of environmental stress. The environment demanded this one. Maybe today it changed, but that is not the case for past 1 million years. It's a constant progress, it's not a one-day process. So we can say that to understand the history, to understand the history of geology or earth back history, just to see today animals. Why this animal is like this? Maybe representing the previous adoptions, its ancestors. Got it? Exactly. So that is one very interesting analogy about genes. So genes are always trying to have this thing. What else interesting genes I can say? So living animal, I give an example. And another example, I said the ant story, right? Do you remember? The ant entire colony acts as a single organism, not a single ant. A single ant will work like a cell in the body. That's why some ants are ready to sacrifice themselves if some snake or anything came to their ant, what we will say, ant hut. Colony, ant colony. When they are coming, their structure, I would say. There might be some English word for that. Anyway, that ant colony. Why some genes, the gene motor is to survival, but why this gene sacrificing itself? Why it is sacrificing not to propagate for the next generation? Why it given instructions to its own vehicle to sacrifice itself? What is the survival benefit of a single ant gene? Here, it's not the survival benefit of a single ant, it's the survival benefit of the entire colony. Can the entire colony be sustained by sacrificing few genes? The motto is to maintain entire colony here, just like our body. Now in our body we have many cells that are constantly dying. That doesn't mean they have death. Their foresight, their entire motto is to sustain this entire colony. Got it? Now we become a, at cellular level, if you think we are alone, actually we are a huge harmonized world of cells working together to be alive actually. Now, next time if you feel alone, don't feel alone. Actually, we are the outcome of that mutual understanding, harmony, love. The cells are loving each other. Some cells are sacrificing just to keep us alive. The greater benefit of other cell colonies in the world. Oh, then I can give another analogy. Next time if you broke up or you have these issues, don't worry, there are one or two people. But as a whole, we all will survive. Don't take such a self-excluding thoughts from this post. That's very easy from genetic point of view. You cannot end your life. You are an outcome of 4.5 billion years of changes. If you are not able to still understand what I am saying, see, I love this evolution actually. Listen, we have a life 4 billion years ago, right? That fish became some reptile, something became mammal. Then suddenly meteorite came, all the dinosaurs died. 99% of life on planet Earth was wiped out. Only 1% survived, right? Now, out of this 1% life, whatever is, during the dinosaur time, Jurassic time, only those 1% again started evolving into the future. Keep in mind, we are the luckiest 1% beings. Now, I just want to tell you another analogy. Today, if you think an ant is a stupid, ant has survived that 1%, that is the luckiest animal, because all 99% died. And we also survived that extinction event. Our ancestors, when I say, not even we, another animal, right? See, we and ant survived because we all have the same ground, that is same earth, same health conditions. So there is no difference between we and other animals. We all played the same game and we succeeded at this game. Got my point? Yeah, just if we think we are great, even the ant is great. Ant is more great because it is still surviving with that small tiny body. Something which we can't survive today. See, next time if you are, even if you are not vegetarians, but still you need to appreciate each other's taste. We are survived. We can, and that is one analogy of seeing genes as an outcome. An immortal coils. With this I will start today's lecture about genes. So this is all about the introduction of genes. This class is 10:20, right? I have 40 more minutes. Fine. So now write down the structure of a gene. Structure of a gene. Okay. So now, a non-coding DNA to come into the gene, there should be some difference, right? Like there should be some written notes or some instructions to state that maybe you are walking in the corridor. Suddenly you came here, this is a gene. How you understood this is a classroom? How would you differentiate a corridor from the classroom? Maybe now you have some entry restricted door. If you are opening this door is an indication of entry into the classroom. Similarly, a gene has three important regions. A gene has three important regions. Write down what are those. It has a promoter region. A gene has three things: promoter 2, promoter region 2, coding region, and 3. Introns and exons. Introns and exons. So a gene has three important regions: a promoter region, a coding region, and a introns and exons. That I will discuss. Now, broadly, these genes can be categorized into two life beings on planet Earth: a single cell life beings and multi-cell life beings. Single cell life beings are called as prokaryotes. As a microbiologist, you have to study prokaryotes because you are dealing with prokaryotes very frequently, such as bacteria, right? So prokaryote and other one is eukaryote. Yeah, eukaryotes. Now let us see. First, let's see what's happening in the prokaryote. So to translation. So first, you know what is the meaning of transcription, right? Transcription and translation. Transcription means conversion of DNA to RNA. So now, first we need to understand from what portion we need to start transcripting. From where we need to convert the DNA to a transcripted RNA. Okay? That will be determined by promoter region. Promoter region will determine from where I need to start my transcription. Translation is a different story. First, let us do transcription to get the RNA piece. RNA will go for coding. Okay. Now, in case of our prokaryotes, the transcription. Wait. So the promoter region in prokaryotes has close to the transcription site. Close to the site of transcription, there is a promoter region. Maybe this is the room starting from here. So we can say from here transcription will start. This is the gene. Transcription will start from this point. To start this one, this is a promoter region, something that you can enter now. This door has a code. They has a label. That label was Pribnow box. This Pribnow box has a sequence, the base pair sequence called TAT-AAT sequence. TAT-AAT sequence in the promoter region of a prokaryote. This Pribnow box is close to the transcription site, and the base pair was TAT-AAT sequence. So they are saying that, I don't know what is this minus 10 and minus 30 box means. Maybe base pair box, you know, from if this is the transcription minus 30 or minus 10. So there are two types of boxes. They are saying that minus 10 box and minus 35 box. Actually, I'm not so good at these things, but maybe there might be some word for minus. Anyway, so minus 10 base pairs and box and minus 30 box. Whereas minus 30 box starts with TTGAGC promoter. TTGAGC promoter. So these are the promoter regions for transcription. Okay? Minus 30 box. Minus 30 box, which is called as TTGACA. TTGACA. Maybe they are the sequences, base pair sequences to initiate a transcription. Okay? So this is for promoter in case of prokaryote. So let us see the promoter in the Eukaryote. So in eukaryote, the promoter region has more, multiple regulatory elements. In eukaryotes, multiple regulatory elements are present that include core promoter and proximal promoter. Here we have core promoter and proximal promoter regions. Proximal promoter regions. So the core promoter regions in eukaryotes, it's a famous one that is called Tata box. TA box. Tata box for eukaryotes. The core promoter region was Tata box, and proximal promoter region is seen in CAAT and GC boxes. CAAT and GC box. So next time when we are sequencing a DNA, if we encounter TATA, that indicates it's a promoter region for some gene. First we got AT, something. Suddenly we got TATA sequence. TATA sequence is always something, the starting of a gene. Okay. CAAT or /GC box. Okay. Just now I recall one interesting fact about life on planet Earth. You know what is the language of understanding to a computer? How many alphabets present to the computer language? Do you have any idea? Only 2 alphabets, only 2 digits, 0 or 1. If you are typing A in a keyboard, it's a sequence of zeros and 1, 0, 0, 1, 1, 0. That is A. B means something, space for something. So entire programming started on bits actually, 0 or 1. And we know the applications of 0 and 1. We thought it's a simple language, right? With 0 and 1, we made supercomputers, our mobiles, laptops, satellites, everything working on 0 and 1. We have understood the benefit of 0, 1. And we have many applications. Just imagine if something having not 2 language inputs, but 4 language inputs now. It will become more complicated than a computer, right? And that's what exactly happening in animals. They have 4 basic language pairs, A, T, G, C and U. So life is more complicated than a computer or a software. Got my point? Because it has 5 letters to write a code. So that's it. This is all about the promoter. In eukaryotes we have Tata box, whereas in the prokaryotes we have TAT, minus 10 base pair box and minus 30 base pair box. That is a TAT, and other one is something WT, Okay, that is all about the promoter. Next one, coding region. Write down the coding region. I just want to know the definition of a coding region from Introns and Exons. Right? What is a coding region? That is one of Exons we have. What is the role of an exact coding region? What it is coding? Of course, it will code gene, but how Exons are different from coding region? They are saying that coding region is a continuous sequence without interruptions. No introns or exons. Okay. Genes are organized into operons. This is for prokaryotes. Let us say in eukaryotes, the coding region has a discontinuous sequence with introns and exons. Fine. Now I understood. See, in case of prokaryotes, the coding region don't have any introns, as I said. Prokaryotes don't have introns. They directly go for genes. So bacteria don't have enough space to maintain the lengthy storage. Okay. So they directly have genes in the circular DNA. Okay. Now, those set of genes in the bacteria, this is gene. The set of genes are located together a slot of genes, a slot of set of genes together called as operons. In case of prokaryotes, write down the in prokaryotes write down coding region is a continuous sequence of genes. Coding region is a continuous sequence of genes without any interruptions, such as introns or exons in a prokaryote. Without any interruptions, such as introns or exons in a prokaryote. These genes sometimes organized, write down, these genes often organized into sets of multiple genes transcribed together, set of multiple genes transcribed together called as operons. O-P-R-A-N-S, operons, operons. So they are saying that in coding region of a prokaryote, they don't have any interruptions, it's a direct set of genes. These genes are interconnected and they will be transcribed together, and this multiple gene set is called as a operons in prokaryotes. Write down in eukaryote now about coding region. Coding region in eukaryote. Coding region in eukaryote. Yeah, definitely this is right now discontinuous sequence with introns and exons. This is a discontinuous sequence with introns and exons. A discontinuous sequence with introns and exons. Write down next point. Introns won't code the protein, whereas exons will code the protein. Introns won't code and exons will code. Write down the similar statement. Introns won't code the protein, whereas exons will code the protein. Or you can write a single statement. Exons will code the protein, whereas introns nothing. Null. Got it? Now, introns are removed. Next point in the eukaryote. Introns are removed during RNA splicing. Introns are removed during the RNA splicing to form mature mRNA, to form mature messenger RNA. So what they are doing, all introns will be removed and they will stitch the exons together, and that stitched exons strand is called as mRNA. Got it? This will be done in eukaryotes but not in pro. So all introns will be spliced and only the genes will be taped together, making a matured mRNA. Now this mRNA will be transferred to the ribosome via a transfer RNA to the ribosome. Got my point? Yeah. Well, next one. Last and final. I didn't want to say much, you already know what are introns and exons. Introns are absent in prokaryotes, exons will code. Same thing in the eukaryotes also. Introns non-coding sequences removed by splicing, whereas exons will be matured to form a final mRNA strand. So far clear about the gene structure? Yeah. Yeah, this is all about the gene structure. In the afternoon, we will discuss other things. By today evening, I will give you complete textbook of molecular biology with all the things that are only present in our syllabus. I am making a textbook and I will give you by evening. Okay? So you can only rely on the textbook, including you will get important questions of 2, 5, and 10 from the same material. I am making some 300 pages material and I will provide this by evening. Fine. Let me take attendance.